This is quite nerve-wracking, actually. Isn't that ridiculous? Because it's just teaching, but... Good afternoon, everyone, and welcome to the NCFM Microbiology Lab. And what I'm going to be doing today is just running through some of the techniques that we do to carry out food microbiology. So I hope you can see me and hear me, and it's very lovely to have you with us, even though I can't see you. And I've decided to call myself for today the masked microbiologist, just because that seems like an appropriate term to use for the situation that we're in at the moment. So as I say, what we're going to do is do some very basic microbiology laboratory work, and what we're looking at today is some sausage meat that's been prepared previously. So I've taken my sausage meat sample and I've stomached it to make it into a liquid solution. And by doing that, I've taken all the bacteria, hopefully, that are within the sausage and mixed it up to create a suspension of those bacterial and yeast cells within this water. So what we're going to do is use these bacteria in suspension. We're going to dilute them down, so in case there are lots of microorganisms in here, we'll dilute them far enough that we'll be able to count them later. So that's the first step. We're going to do a serial dilution. The next step then is we're going to do some spread plating onto a nutrient agar, and this will allow us to see the types of organisms that will grow just on a good general growth medium. So this is looking for the bacteria within the sample. And then we're also going to carry out some pore plating using VRBGA agar, and that will allow us to look for the presence of Enterobacteriaceae in our sample, which are a really common indicator of fecal contamination within our product. So let's get started. First, most important thing to do in the microbiology lab is to get our Bunsen burner going so that we've got a nice sterile environment to work with. Now, one of the problems that we have with videoing and recording with a Bunsen burner on is the Bunsen burner is very noisy. So although I should have it on as a roaring blue flame all the way through the process that I'm doing, I'm going to end up turning it up and down so that we don't have the constant noise of the Bunsen burner in the background, because we don't want that to distract what we're doing. So let's get my Bunsen burner on first. We're going to turn it up just a little so that it's on a blue flame but not too noisy. So one of the most important things in the microbiology lab is that everything is labelled, so we know precisely what we're doing and we can go back and find all of the bottles and solutions and everything else that we're working with. So hopefully you'll be able to see that I've already labelled up my stomached sample. I've labelled it up here as sausage with my initials and the date when I set that up, which was today. Now, the first thing I'm going to do is a serial dilution. So I have 5 universal bottles here, so these are small glass bottles, and in 4 of those I have 9 millilitres of quarter strength Ringer's solution. And quarter strength Ringer's solution is used as a diluent. So that means we can dilute our sample down, and the organisms, when they're in this diluent, won't be damaged. They won't burst open because there's too much water in there, for example. This is a salt solution, which keeps them in a nice osmotic balance. So we're going to label these up first. And first, the original sample, I'm just going to label as a 10 to the minus 1, because previously when I made this up, I used 10 grams of sausage and about 90 grams worth of water to make a 1 in 10 dilution. Then the next bottles in this series are going to be minus 2, minus 3, minus 4, and minus 5. And I'll explain why I'm calling them those as I go along. So the first thing I need to do is try and get my sample out of this bag, because this is big and messy and dirty, And what I don't want to do is end up getting bits of sausage meat all over our lab equipment. So I've taken an empty, sterile glass bottle, the one that I've labelled as 10 to the minus 1. And I'm just going to sterilise the neck of the bottle and then transfer just a little bit of this liquid into this bottle. It'll be a lot easier to use then for all of the subsequent steps that I need to do. Okay. Again, flame the neck of the bottle, put the lid back on, and back into the rack for now. So this sample is now finished. Just going to wrap it up and put it to one side. And this will go for autoclaving later, so if there are any harmful bacteria in here, then we destroy them in that autoclaving process. So here we go. Here's our original sample. And what we're going to do now is take 1 milliliter of this sample using an Eppendorf pipette, which is set to dispense 1 milliliter or 1,000 microliters. I'm going to take 1 milliliter of this sample and place it into 9 milliliters of Ringer's solution. And that will make a 1 in 10 dilution of the sample that we're working with. So I'll take a clean tip. Again, flame the neck of the bottle. Slowly withdraw 1 milliliter of liquid. Put that bottle down. Open up my 10 to the minus 2 dilution. Dispense the liquid in. Discard the tip, and again, flame the neck of the bottle. Now, the reason I do all of this work around the Bunsen flame and flaming the neck of the bottle is to make sure that we keep everything sterile and clean, partly to protect the samples, but also partly protect me from any microorganisms that we don't want around. So I now have a slightly diluted sample. You can see it looks a bit less cloudy. I'm going to then take this 10 to the minus 2 dilution to create a 10 to the minus 3 dilution. So again, same process. Flame the neck of the bottle, withdraw 1 milliliter, put that bottle away, pick up my 10 to the minus 3 bottle and dispense the liquid in again. Now what I've done here is create another 1 in 10 dilution, so 1 milliliter into 9 milliliters, which makes a 1 in 10. But because we took that from this 1 in 10 dilution, what we've ended up with is creating a 1 in 1,000 dilution when compared to our original sample. And 1 in 1,000 is also known as 10 to the minus 3. So now what we can do is just continue that series down, and I'm going to keep going until I reach a final dilution of 10 to the minus 5. And that, I'm sure, will be enough of a dilution to make sure that I can count and visualise all the organisms that are present in my sample. So transfer the liquid, give it a good shake. And one final time, draw 1 milliliter. Dispense in the bottle marked 10 to the minus 5. Give it a good shake. And our serial dilution is now completed. So now I have this dilution of microorganisms from 10 to the minus 1, which was the original food sample that I prepared through stomaching, minus 2, minus 3, minus 4, minus 5. What should happen in this dilution, of course, is the number of bacterial microorganisms in each should be 10 times fewer at each step of dilution. So let's say we have 1,000 microorganisms in here per milliliter. We'll have 100 in here, 10 in here, 1 in here. And we'll be able to measure that then by putting these microorganisms onto microbiological growth media. And the first one we're going to use is nutrient agar. So nutrient agar is a really good general growth medium, and it allows almost all types of bacteria to grow, and quite often some yeast, some fungi as well, even though that's not what this is particularly designed for. So by transferring the liquid from our dilution onto these plates, this will allow us to calculate what is known as a total viable count, or aerobic colony count, of the food that we're looking at. And that's one of the most important counts in terms of microbiological specifications for food, as it gives us a good overall estimation of how contaminated the food product is. But again, before we do that, it's really important that we label what we're working with so we can go back and find out exactly what we're looking at. So I'm going to put on each of these plates my name and the date, and then also the sample, which in this case is sausage, and the dilution factor, which is 10 to the minus 1. Ten to the minus 2. Ten to the minus 3. To the minus 4. And finally to the minus 5. Now what you'll notice as I did that was I wrote as small as I could and as close to the edge of the plate as I could, because then by doing this, when I see bacteria growing on the surface of the plate, they won't be obscured by lots of writing. At least that's what we hope. So what we're going to be doing in this case, it was a bit noisy there, wasn't it? We're going to be pipetting 0.1 millilitres or 100 microlitres onto the surface of the agar plate, and then spreading that liquid around. So we spread the bacteria right the way across the surface of the agar, and then they'll go in the incubator, and we'll be able to look at the results of those tomorrow. So I'm going to take this time a 100 microlitre pipette, which is set to dispense 100 microlitres of liquid, and this time we're using the smaller tips, the yellow tips, for that to happen. So we'll start with 10 to the minus 1, take our sample again, just give it a quick mix. Flame the neck of the bottle, withdraw 100 microlitres. It's always a bit tricky on this one because of bits of meat. Flame the neck again and place it down. And then I'm going to dispense this liquid onto the surface of the agar, nice and gently. Next I'm going to take a plastic spreader, so these are sterile. Pick up sticks sometimes, but there we go. One of the spreaders, and then I can use this to smear that liquid right the way across the surface of the agar, making sure I get into all of the cracks and all of the edges to spread it as effectively as I can right the way around. Spreader then goes into the waste discard for sterilisation, and that plate is then ready for incubation. So I'm just going to carry that on. So again, 100 microlitres, this time of my 10 to the minus 2 dilution, onto the surface of the plate. Take the spreader. Ten to the minus 3. And then finally 10 to the minus 5, which is the most diluted sample. And those are all my spread plates put together. Now, what's going to be important with this when we go in for incubation is that we put the plates so that the agar is at the top. And that's really important because the plates will get warm. We'll be incubating these at about 30 degrees centigrade. And what will happen is that the plates will start to sweat a little bit, and obviously the microorganisms as well will respire. And if we have the plates the other way around, you can see actually already in this case, hopefully, if we switch it, good, you'll see there are some droplets of liquid on the surface. Is that better? Yeah? Some droplets of liquid on the surface of the lid. And if we leave it this way, those droplets will get bigger and then drop onto the surface of the agar, and that can cause our plates to look very messy after incubation. So those are all set and finished now. What we'll do is put those in the incubator, which is in the other room a bit later on, and then we can have a look at those in about 24 and 48 hours to see what we have growing on them. And of course, as I mentioned earlier, what we're hoping to see is a dilution where there are 10 times more bacteria as we go up through the dilution series. So if we see, say, one organism on the 10 to the minus 5 plate, we'll expect to see 10 on the 10 to the minus 4, 100 on the 10 to the minus 3, and then probably an uncountable number. There should be about 1,000 on the 10 to the minus 2 plate, which we wouldn't be able to count. So that's spread plating. And then the last technique we're going to look at today is a slightly different type of technique, but again using microbiological growth media. And this is a technique known as pore plating. And what we're going to be using with this is a molten media called VRBGA. And this medium is really good for the growth of Enterobacteriaceae. So these are organisms like E. coli, which grow within the gastrointestinal tract of animals. They also, though, are really dangerous in terms of food poisoning. So if we have lots of Enterobacteriaceae in our product, it may be a cause of food poisoning for our consumer at the end. So it's useful for us to check and see what concentration of these types of organisms we have in our samples. Now, the reason we're doing pore plating with these is a lot of these organisms like to grow in slightly less oxygen. So if you put them on the surface of a plate, they won't all necessarily grow. What we're going to do is mix our organisms in with the molten agar, allow the agar to set, and then incubate them. And that means some of the bacteria will actually be embedded within the agar as it sets. And that means they've got slightly less oxygen, so they'll be happier in terms of growing, and we'll get a more accurate count of the number of organisms that we have in there. So I'm going to label these plates in exactly the same way. Again, with my initials, date, the type of sample we're working with, and the dilution factor. The other thing sometimes you might need to consider is labelling up your plates, if they haven't already been done for you, with the type of agar that they contain. Now, it's very easy for us today. Our nutrient agar is beige colour and our VRBGA is purple, so we're not going to get those mixed up. But actually a lot of microbiological growth media are very similar beige colours, so it's important that we know which is which so that we can identify them. So sometimes we do need to label our plates with the different colours as well. Now this time, because we're using a pore plate and we're mixing our sample in with the agar itself, we can actually put more inoculum in there. We can use a whole milliliter of our dilution series this time. And that's really helpful because it allows us to count the number of microorganisms in a larger volume of liquid, which means we're more likely to be able to detect them. So I'm going to go back to using my 1000 pipette, so set up to measure 1000 microliters of liquid, and the slightly larger tips again. And this time I'm going to put our dilution directly into the empty Petri dish. So withdraw 1 milliliter of my sample, dispense it into the empty Petri dish. And I'm going to do it slightly differently this time. I'm going to set up all of my dishes first before I work with my molten agar. So 1 milliliter of my 10 to the minus 2 dilution. To the minus 3. Oh, fingers aren't working. That's on the minute. 10 to the minus 4. And finally, 10 to the minus 5. So now I have my inoculum in all 5 of my empty Petri dishes. I'm going to pour the agar onto this. So this is nice and cool now. I've just heated it up, but I've left it out for a bit. So it's now cooled down to about 50 degrees, which is really important because if it's too hot, when we pour it onto our samples, we're going to start killing off the bacteria that are present in there, which obviously we don't want to do. But this is now cool enough to pour. So I'll be flaming again the neck of the bottle, and I'll pour about 15 milliliters of this agar into the plate. And just give it a really good swirl. So first in one direction, then in the other, and again finally back, and then move that out of the way. And those pour plates will now, oh, there we go. Those pour plates will now set. They'll set quite quickly, the agar. When it gets down to about 35 degrees, the agar will start setting. So we need to leave those just for a little bit in this orientation to let them set properly. And then when they have, we'll again turn them upside down so the agar is on the top, and put these ones in for incubation at 37 degrees. So obviously, because we're looking for Enterobacteriaceae, which live in the guts of animals, including humans, they really like to grow at about 37 degrees. So that's the best temperature for them, and we'll be able to see those results tomorrow to do counts of how many organisms we have within our samples. So that's everything I wanted to show you today. Thank you very much for joining us. I hope you've enjoyed it. Do please give us some feedback and let us know how it's gone. And I'll look forward to seeing you all soon. Thank you very much.